Welcome to Evolutionary Biology. Today, we are exploring a fascinating topic called chemogeny. Imagine trying to bake a complex, delicious cake without a baker. Just raw ingredients flying around in a room, waiting for a lightning strike to perfectly bake them. It sounds like magic, but this is exactly how life began on our planet billions of years ago. We will uncover how the universe baked the very first ingredients of life from completely lifeless chemicals. Now that we have pictured this magical baking process, let us define what it actually is. Chemogeny, or chemical evolution, is the scientific theory describing how simple, inorganic molecules naturally combined to form the complex organic building blocks of life. The word comes from chemo, for chemical, and geny, for creation. According to the famous Oparin-Haldane hypothesis, the early Earth had a reducing atmosphere. This means it completely lacked free oxygen. Instead, it was filled with simple gases like methane, ammonia, hydrogen, and water vapor. Understanding this environment is crucial because it sets the stage for the chemical reactions that made life possible. Building on that reducing atmosphere, we must ask how these simple gases transformed. Under the intense heat of ancient volcanoes and the violent energy of massive lightning storms, these simple gases reacted with one another. They formed the first amino acids, sugars, and nucleotides. These heavy organic molecules eventually rained down into the oceans, creating what we call a rich primordial soup. This concept bridges the seemingly impossible gap between a dead, rocky planet and a vibrant, living world. Furthermore, this is the exact foundation of modern astrobiology. When scientists send rovers to Mars or Titan, they are actively looking for these exact same chemical precursors. Knowing what formed in the primordial soup, we must now look at the physical rules that governed these changes. To truly understand how these lifeless chemicals combined, we have to look at the rules of thermodynamics. On early Earth, forming complex molecules from simple ones required fighting against the natural tendency of the universe to become more disordered. Normally, chemical reactions want to increase chaos, not build highly organized structures like amino acids. This means that the violent conditions of early Earth, like extreme heat and lightning, were not just random weather. They were absolutely necessary energy inputs required to force these life-building reactions to occur against the normal flow of thermodynamics. This thermodynamic struggle brings us to a fundamental mathematical relationship. We can understand whether a chemical reaction will happen naturally by looking at the Gibbs free energy equation. The formula states that delta G equals delta H minus T times delta S. Let us break down these symbols. Delta G represents the change in Gibbs free energy, which tells us if a reaction can happen on its own. Delta H is the change in enthalpy, or the total heat absorbed or released. T stands for the absolute temperature, measured in Kelvin. Finally, delta S represents the change in entropy, measuring the chaos in the system. For simple gases to combine, entropy decreases, making delta S negative. This normally makes delta G positive, stopping the reaction. But the extremely high temperature T provided the energy to overcome this barrier. Let us apply this formula conceptually to the conditions of early Earth to see the problem. We are given that simple gases are combining into complex organic molecules. This means the disorder, or entropy, decreases. Therefore, delta S is negative. In our formula, subtracting a negative T times delta S turns that term positive. This means the overall delta G becomes positive. As we learned, a positive delta G means the reaction refuses to happen on its own. This step-by-step breakdown physically proves why the massive energy inputs from lightning and extreme heat were absolutely mandatory. Without that massive external energy, the mathematical barrier of a positive delta G would have permanently prevented the origin of life. Now that we understand the thermodynamic energy required, let us look at the sheer mathematical odds of these molecules assembling. Imagine the early Earth's primordial soup, filled with various simple amino acids floating around randomly. We want to find out the exact probability of these amino acids bumping into each other to form a specific tiny protein chain. Since the amino acids link up purely by random chance, the probability of getting the correct amino acid at each position in the chain is completely independent of the others. We need to calculate how likely it is for the universe to build a specific chain purely by accident, showing us the massive mathematical hurdle that early Earth had to overcome. To calculate those odds, we use the basic rule of independent probabilities. The formula is P equals the fraction 1 over n, all raised to the power of L. Let us understand what each variable means. The variable P represents the total probability of forming our specific target sequence of amino acids. The variable n represents the total number of different amino acid types available in the primordial soup. The variable L represents the length of the polymer chain we are trying to build. Physically, as the length L increases, the probability P drops drastically. This mathematical relationship perfectly illustrates why building long, complex proteins by pure chance is incredibly difficult and requires vast amounts of time. Let us apply this probability formula to a concrete scenario from the ancient oceans. We are given that there are exactly 10 different types of amino acids available, so n equals 10. We want to build a chain that is exactly 5 units long, so L equals 5. We substitute these known values into our equation. We replace n with 10 because there is a 1 in 10 chance of the correct amino acid attaching at the first position. We replace L with 5 because this lucky attachment must happen perfectly 5 times in a row. Calculating the exponent gives us 1 over 100,000, or 0.00001. This incredibly small number physically means that only 1 out of every 100,000 chains will be the exact sequence you want, proving why Chemogeny required millions of years and entire oceans of water. Having seen the mathematical odds, you might wonder if we can actually prove this happened. In 1953, scientists Stanley Miller and Harold Urey did exactly that. They built a glass apparatus to physically simulate the chemogeny of early Earth in a laboratory. They filled it with the simple gases of the reducing atmosphere and ran continuous electrical sparks through it to simulate ancient lightning storms. After running this setup for just 1 week, they collected a pool of liquid at the bottom, representing the primordial soup. This was a groundbreaking moment in evolutionary biology because it physically proved that you do not need living cells to create the building blocks of life. Let us look closely at the actual apparatus Miller and Urey designed. On the bottom left, you see a flask of boiling water, representing the ancient hot ocean. The water vapor travels up into the large upper flask, which contains the simulated atmosphere of methane, ammonia, and hydrogen. Notice the electrodes inserting into this chamber? They are firing sparks to simulate the violent lightning of early Earth. As the gases react, they are pushed down through a cooling condenser, representing rain. Finally, the condensed liquid collects in the trap at the bottom right. This trap represents the primordial soup, and this is exactly where the newly formed amino acids were collected after just 1 week of continuous operation. To understand the results of that experiment, we need to measure exactly how much material was created. Chemical reactions depend on the number of molecules present, not just their raw weight. Therefore, we use the formula N equals M divided by M to convert raw mass into moles. Let us define these terms. The variable N represents the number of moles synthesized by the artificial lightning. The variable M is the given mass of the substance collected, measured in grams. The variable M is the molar mass of the specific chemical, which is a known constant value in grams per mole. Physically, if the mass M of the collected sample increases, the number of moles N will also increase proportionally, giving us a direct count of the molecules forged. Let us apply this formula directly to the yield from the Miller-Urey experiment. We are given that the scientists collected exactly 0.075 grams of the amino acid glycine, so our mass M is 0.075. The molar mass of glycine is a known constant of 75 grams per mole, so M equals 75. We are asked to find the number of moles. We substitute these values into our equation, dividing 0.075 by 75. This calculation gives us exactly 0.001 moles of glycine. This step is crucial because moles give us a direct, standardized count of how many glycine molecules were actually forged by the electrical sparks from thin air. Now that we have the number of moles, we must determine how concentrated this primordial soup was. We use the molarity formula, which is C equals N divided by V. Let us look at the variables. The variable C represents the molarity, or the final concentration of the solution. The variable N is the number of moles of the substance we just calculated. The variable V represents the total volume of the liquid trap, measured in liters. This is the standard way chemists measure concentration. Physically, if the volume V were smaller, the concentration C would be much higher. This inverse relationship is exactly why scientists believe life may have started in shallow, evaporating tide pools, where the volume was constantly decreasing. Let us finish our analysis of the experiment by calculating this final concentration. We know from our previous step that the number of moles, N, is 0.001. We are given that the water trap has a total volume V of 1.5 liters. We are asked to find the molarity, C. We substitute these values into the equation, dividing 0.001 by 1.5. This gives us a final concentration of approximately 0.00067 molar. While this number might look very small, it is actually a massive and groundbreaking result. It physically proves that just 1 week of simulated early Earth conditions can generate a highly measurable and significant concentration of amino acids. We have reached the end of our journey through the chemical origins of life. Let us reinforce the most important ideas. First, chemogeny is the magnificent prologue to life, where simple inorganic gases were forced to combine into complex organic molecules. Second, we saw mathematically how the extreme energy of a violent young planet was absolutely necessary to overcome thermodynamic barriers and tiny probabilities. Finally, The Miller-Urey experiment proved that this is not magic. It is a mathematically probable series of chemical reactions. You have now mastered the chemical foundation of evolutionary biology. This perfectly sets the stage for your next great leap in understanding: the formation of the first self-replicating RNA molecules.